An NIAMS researcher is seated outside in front of a building speaking to the camera. Robert Kwiat, Functional Immunogenomics Section, NIAMS.
RNA molecules have many functions in biology, and when scientists are trying to understand the function of a new RNA molecule, an important question to ask is where in the cell that RNA is primarily located. Is it in the cytoplasm or is it in the nucleus? The cell you see here is called a monocyte.
The view changes to a computer-animated image of a single cell, rotating slowly. Cellular localization of specific RNA molecules by HCR, primary human monocyte. Parts of the cell are labeled: nucleus, plasma membrane.
Monocytes play an important role in the body's defense against microorganisms and toxins. This cell was extracted from the blood of a healthy human donor.
The camera circles around the lens of a microscope.
These images were obtained using a type of microscope called a confocal microscope.
It cuts to a view of the researcher operating the equipment from a computer. The animated rotating cell is shown again.
The cell membrane is stained in red and the nucleus is stained in blue.
The researcher is at his lab bench preparing a sample with a pipette.
Using a technique called hybridization chain reaction, or HCR, we will find where the specific RNA is found within the cell.
The computer image zooms inside the cell. The camera pans across the surface of the nucleus.
Zooming into the cell, in the cytoplasm, we can see the RNA in the form of a green glow.
Computer-generated spheres at areas of high signal.
We use a computer program to mark spheres at areas where the RNA is found. This makes them easier to see. If we zoom out, we see the nucleus of the cell,
Cytoplasmic transcript localization,
and the location of transcripts in the cytoplasm outside the nucleus. Now, if we zoom into the nucleus, we can see RNA located there.
nuclear transcript localization. The nucleus spins on the screen. The nucleus becomes transparent as the camera zooms in, revealing red spheres inside.
We can see that this particular RNA is much more abundant in the nucleus than in the cytoplasm.
The researcher is now seated inside a building by a window, speaking to the camera.
This information gives investigators a first clue about the types of functions this RNA might have. It also helps them choose the best techniques to further study this RNA in future experiments.
Logo for NIH, National Institute of Arthritis and Musculoskeletal and Skin Diseases.